Good afternoon, Dr. Muyi. How are you doing?
I'm fine. So we are
Yeah.
welcome to today's session on microbiology meets data science. And this initiative is called the Classroom to Careers Initiative, and where our aim is to help as much microbiologists, biological scientists to be able to learn and to be able to have ideas of career path. The goal is to take microbiology away from the regular things that we know exist, because there are many more opportunities that we can explore as microbiologists. My name is Moyelu Akitunde. I'm currently doing a postdoc in Germany, and I am an industrial microbiologist by training. And my colleague today is also an industrial microbiologist by training, our speaker for today. And we've we met a long time ago during our master's degree in UI. So she's someone that has delved so much into bioinformatics, data science, and successfully combined both aspects of microbiology. For us, whether graduates, undergraduates, those that are employed, there's so much that we can gain. And there's so much that we can do with our microbiology course of study. So I'll give myself as a very good example. Currently, as a postdoc, I am working alongside mechanical engineers. I'm working in an institute for sustainable polymer materials. However, I am studying biodegradation of polymers. So we are talking about polymers used in engineering applications, automobiles, aerospace, and the likes. So, as microbiologists or biological scientists, we see that we are functional and we can function in as much places as possible. So, I want us to be open minded and learn as much as we can from our speaker for today. So, before we start, because I don't want us to waste time, yes, we can give it some more time for more people to join in, but I believe that we we would have questions, and we would have people want to ask more questions about bioinformatics, data science, and the likes. So I'm going to read the bio of our speaker to us today, and then I would give her the floor to break data science to us, because I'm also here to learn so much from her. So our speaker for today is Oluwabukola Atinuke Pukwola. She's an industrial microbiologist. She's a data scientist, and she's an industrial microbiologist, a data scientist, and a genomist. And the interesting thing is that I like the fact that she's an industrial microbiologist. That's the first thing. And she's had over 13 years of experience in visual analytics, data analytics, and bioinformatics. So I believe she's going to really tell us what visual analytics is, what data analytics is, and what bioinformatics is. Her work lies in the intersection of microbiology, genomics, and data science, where she applies computational and experimental approaches to explore alternatives to antimicrobials. She focuses on antimicrobial peptides and microbial biotechnology with applications that spans food systems, medicine, pharmaceuticals, and agriculture. She's committed to using genomic and data-driven insights to drive innovation in public health, sustainable biopreservation, and biotechnology. And she's a research officer in the departments of genetics, genomics, and bioinformatics at the National Biotechnology Research and Development Agency, NABDA, in Abuja, Nigeria. So I want us to welcome her as she takes the floor and presents to us and speak to us about data science and microbiology. Over to you, Ma.
Yeah, so good evening, Dr. Moyoluwa Akintunde. It's a pleasure sharing this floor with you this very evening, this 25th of September 2025. I'm so happy to be in your midst in this very evening. Yeah, so like she said, that we met long time ago. That was when we were in the University of Ibadan in the unit of industrial microbiology. Yeah, so over time we dispersed and we've been connecting. Thank you for the power of social media. Yeah, so I think 1 month ago when she reached out to me that she would love me to come to your midst and talk about where microbiology meet data science. I didn't have any choice than to say, well, amidst our busy schedule, I just have to do the needful. So I welcome everyone, whatsoever your background is, whether you're a biological scientist, whether you are a physical scientist, a chemical scientist, and you are just trying to look around you and what's this feels of all about data science. Relax yourself. There's no big deal. So we are here today to be able to shed light on how biological science, microbiology, biosciences can actually intersect with data science and give insight into our research work. Yes. So people are beginning to ask different questions, like in my alumnus group, people were asking that is microbiology becoming obsolete? Can I share my screen? Let me share this screen with you now.
Yes, you can. You
Um,
can.
okay, let me share this screen so that we can be able to flow along. Yes, can you see my screen? If you can see it, kindly let me know.
Yes, we can.
Okay, so in my alumnus group, like I said, they're beginning to deliberate and discuss that is microbiology becoming obsolete in today's world? But they were like, after spending 4 years in school, they got out to be met with the realities in the labor market. A microbiologist that came out that want to diversify to probably medicals or agriculture or pharmaceuticals, and they're like, no, you didn't have the requirement, or even food industry. They're not like, is microbiology sufficient? Is it an obsolete course? So we are here this evening, whether you're a microbiologist, a biological scientist, to open our eyes to see how far we can go. Yeah, so I tag today's presentation, Microbiology's New Frontier, powered by data science. Like you have heard, I'm Olua Bukola Tinike Popola. I work at the intersection of microbiology, genomics, biotechnology, and also data science. I'm a research officer at the National Biotechnology Research and Development Agency under the Federal Ministry of Science and Technology in Abuja. So join me as we look through where microbiology meets data science. Yes. So, okay, I think I need to project as, yeah. Did I see the full screen? Can you see my full screen in the presentation? Okay, yeah. Mode.
Yes, we can.
Yes. So we are looking at the evolving frontier of microbiology in an era that's essentially masked or defined by vast data streams. We are beginning to ask a question: that is traditional microbiology, the conventional microbiology, is it facing obsolescence, or is it poised to evolve into an even more powerful and indispensable scientific discipline? Let's put it in a layman language. Is microbiology becoming obsolete in this age of big data, or is it evolving into something more powerful? Let's look at it together. Because at this period of time, microbiologists across the nations of the earth, especially in Africa, the low resource settings, the low and middle income countries, LMIC are beginning to ask, is microbiology worth studying? So why this topic matters now more than ever? Firstly, because of the biological data explosion. The rapid increase in genomics, proteomics, and microbiome studies is generating a lot and large data volumes, you know, creating a need for new methods of analysis. That is where data analytics, data science comes in. And the new analytical skills, that we require to be able to analyze these complex data sets, complex biological data sets. You can agree with me that in your laboratories, a lot of data are being generated every day in the wet laboratories. How do we transform this raw data into actionable insights? You're just looking at a whole lot of dirty data, gigantic data in your Excel sheet, in your Microsoft Word, a lot of different charts, different diagrams. You are looking at how do I make sense of this data. That is why this topic is very important now more than ever. And also the evolving job markets. Life sciences roles are increasingly requiring data-driven competencies. Everybody wants to make sense of the data they are generated, because you are generating data in your wet laboratory, physical laboratory, chemical laboratory, biological laboratory, molecular biology laboratory, even polymer laboratories, peptide synthesis laboratories. A lot of data are being generated. People are worried. The employers are worried. They want to actually make sense of this data. They want to be able to infer inferences from those data that are coming out of those laboratories, shifting the landscape of traditional microbiology careers. Microbiology meet data science. Where do they meet? In unleashing the future. I welcome every one of you to this journey where the microscopic world of microbiology that you learned about in school converges with the expansive universe of data science. Microbiology is such that they are tiny organisms that your eyes cannot see, your naked eyes cannot see. You require microscope to be able to view who they are, what are they doing there, So this presentation solely aims to illuminate how our conventional biological research, microbiological research can be revolutionized and amplified through the power of data that you as a scientist are generating, ensuring its continued relevance and innovative potential. The evolving landscape of biological science. What are we trying to do? We are trying to bridge disciplines. Microbiology is not becoming obsolete. That's the good news for you. It's evolving. We are integrating data science, to provide powerful new tools for discovery and application, rather than for you to dump your data on the shelf in your thesis, in your reports, in journals, in manuscripts. We are going beyond dumping those data into making powerful insights from those data that you once dumped on your shelf. We are creating new avenues for innovation. From genomic insight to predictive modeling to data science, we are unlocking novel pathways for understanding and manipulating biological systems. Your microorganism is a living entity, like very tiny, minute, microscopic. And funny thing is that they are ubiquitous. They are present everywhere, in the air, in the soil, in your sputum, in your blood sample. They are there in your gut. They are there inside your food. They are there in your plants. What are we trying to do? Apart from isolating them, characterizing them, viewing them in the microscope, we are looking at their biological systems. How are they partnering with plants? How are they partnering with humans? How are they partnering in your food system? What are they doing there? The advox, the good that they are doing. I remember one course that we took, I think in my MCB 201, microbes as friends and foes. We want to look at the applications, in real life, to predict from their system, what are they doing, from their genes. We're also trying to look at the enhanced impacts, by embracing data-driven approaches. We want to accelerate research. We want to develop targeted solutions. We want to address global challenges more effectively, looking at the SDGs. Now, I've been speaking generally, I'm narrowing down to my own journey from the lab bench to data insights. Like Dr. Moyi said when she was reading my bio, that my research focused on antimicrobial peptides. You know, these are small molecules, biological molecules, ribosomally synthesized by microorganisms, leveraging a unique interdisciplinary background spanning industrial microbiology, genomics, biotechnology, and data science. I've been looking at antimicrobial peptides from bacteria. These peptides are also present in plants. They are present in diverse environments, you know, that have the ability to inhibit or kill other microorganisms. Yes. So antimicrobial peptides, they are like nature defense system. In the where they are originally found, they are like innate immune response of those microorganisms. They are actually used as their own defense system to be able to compete in the environment that they are found. Like I've said, that they are small, naturally occurring molecules with broad spectrum antimicrobial activity. They represent a promising alternative to traditional antibiotics and synthetic preservatives. They are produced by various organisms, bacteria, even humans produce them as part of the natural of their innate immune response, you know, to respond to environmental factors. They have broad spectrum activity. They are effective against bacteria, fungi, viruses, even some parasites, you know, with novel mechanism of action like, you know, pore formation. They shut down the RNA or DNA synthesis of some microorganisms. They have diverse mechanisms of action. And one thing about them, very, very important, uh, very, very important activity or very important uniqueness about them is such that they have limited resistance. They are less prone to resistance development compared to the conventional antibiotics, which is very crucial in this era that we are combating antimicrobial resistance globally. The challenge, one of the challenges that we encounter is actually how to discover these novel antimicrobial peptides. Because the conventional methods of AMP discovery are often time consuming and resource intensive. And in this era or in this climate where there is limited resources for research, data science offers a paradigm shift. If you look at this chart on the screen, the traditional or the conventional AMP discovery involves random screening, you know, getting these microbes in vitro, testing them against different arrays of pathogens or spoilage microorganisms. There's slow validation cycles because you have to go to the laboratory, weeks, months of intensive wet laboratory work. But here comes data science. With the advent of genomics, the large data already available in public domain, the NCBI, the Assemble, the Uniprot, a lot of data have been dumped online. Anti-Smash, we have Badge, they are all online, you know. Data science enables us to have genomic prediction of these antimicrobial peptides from the genomes of these already sequenced database available online. We are able to have rapid prioritized testing because once we are able to quickly profile these antimicrobial peptides, we bring out their genes, the chromosomal cassette, we see the biosensitivity pathways. We are able to quickly identify the genes, even the location of the genes responsible for the production of these novel antimicrobial peptides. Rather than for you to go through the conventional microbiological method of isolation of the microorganisms, of characterization, screening, biochemical tests, physiological tests, and all of those. Data-driven AMP discovery is a rapid methodology of actually discovering novel AMPs. So the aim is to overcome this limitation by harnessing the genomic data to predict and validate novel antimicrobial peptides efficiently. I'm only using antimicrobial peptide as a case study. Any area that you're working on too, we can narrow it down to and navigate. I only want to drive this point home for you to be able to, understand what we are talking about, how to be able to intersect microbiology with data science. Data mining genomics for AMP prediction. Our approach is the integration of cutting-edge data science techniques with vast genomic data sets to identify potential antimicrobial peptide candidates using genomic data collection, gathering comprehensive genomic and metagenomic data from diverse microbial sources. For instance, I work on bacteria, for instance, now antimicrobial peptide producing bacteria that we are identified that we are already isolated and characterized in the microbiological lab. They were already tested to have in vitro broad spectrum activity against pathogenic and spoilage microorganisms like Pseudomonas aeruginosa, Escherichia coli, Staphylococcus aureus, Bacillus cereus, and the Salmonella typhi. And those on have broad spectrum activity, their sequence have been obtained. They've been done, 16S sequencing, whole genome sequencing, blasted on NCBI, and some of them actually promising novel strains, and we go forth for bioinformatics analysis using algorithms, machine learning models to scan for AMP-like sequences and features. And interestingly, we are already seeing novel antimicrobial peptides. And we use predictive modeling. We develop models that forecast antimicrobial activity and potential efficacy based on sequence characteristics and the experimental validation, synthesizing predicted AMPs and testing the antimicrobial peptides in vitro and in vivo using animal models. You can imagine sitting down in the laboratory for like 6 months, 1 year, 24 months, 18 months, working on in the wet laboratory. You are profiling, analyzing data. You are actually isolating, characterizing microorganisms. But within few weeks, you go down to your dry laboratory, you are able to see the fingerprints of those organisms live on your screen with the aid of visual analytics, data science, bioinformatics, predictive modeling, genomic analysis. You are seeing the amino acid content of those organisms, you are seeing their proteins, you are seeing the biosynthetic pathways, you are seeing the gene clusters, you are seeing the genes coding for specific activity that you have already seen in vitro. You're beginning to look at, wow, look at this. The power of multi-omics integration. Beyond genomics, integrating multi-omic data provides a holistic view, revealing deeper insight into AMP function and interactions. Even beyond AMPs, whatsoever you want to lay your hands on, your area of research, genomics, proteomics, metabolomics, interactomics, they have been able to narrow down for us how to go directly. You narrow down on what you want to achieve. The genomics is talking about the blueprint, the DNA sequences and gene predictions. You're looking at the genomic component, genomic map, or the particular organism that you're working on. You're seeing the chromosomal cassette. You're looking at the location of the gene of interest, to prepare you probably for CRISPR-Cas, for genomic, for cloning, for gene editing, for genome editing. You're looking at proteomics, the workers, people that are working, the protein expression, how are they expressed, the function. Like I saw the antimicrobial peptides, they are proteins. They are short chains of amino acid less than 50. You are seeing the way they are expressed. You are looking at the numbers. You are looking at the location. You are looking at where they are present on the chromosomal cassette of your organism. Interotomics, the network, the molecular interactions, the pathways, how those enzymes, how are they crossed, how are they expressed. I saw some azole rib in one of my studies. That's azole. They are like heterocyclic ring. Now, after those peptides are synthesized, They are post-translationally modified with that iso ring, making them stable. And I've seen that in vitro, they were able to withstand it up to 100 degrees Celsius. They were able to withstand pH, both low and acidic pH, wide range of pH. These isolates were able to, you know, they were susceptible to proteases, different enzymes, proteolytic enzymes. And I saw those molecular interactions and pathways when we're doing genomic analysis. Metabolomics, the product, the metabolic pathways, the small molecules. You cannot see all of this in your wet laboratory. You can't see all of this when you're doing microbiological work. So microbiology is not becoming obsolete. It provides for us a framework, the data, to be able to utilize to go further into genomics, proteomics, interotomics, and metabolomics. Very interesting course. What are the diverse applications of novel antimicrobial peptides? The discovery of novel antimicrobial peptides has far-reaching implications across multiple sectors, offering sustainable and effective solutions to food systems. They serve as natural preservatives for enhanced food safety and extended shelf life, alternative to synthetic chemicals, alternatives to antibiotics. In medical and pharmaceuticals, they are new therapeutics agents against antibiotic-resistant infections. They are anti-cancerous, they are antiviral, a whole lot of different applications. In agriculture, they are positioned for sustainable alternatives to chemical pesticides and growth promoters. Behold, novel antimicrobial peptides to the world. And these are embedded in those tiny microorganisms that in the laboratory, you have already finished them, you have put them on plates on your slant, you don't know what next to do with them. Whereas they are carrying potential footprints of a lot of things, of enzymes, of antimicrobial peptides. The power of the combo, microbiology, where it's meets data science. The synergy between microbiology and data science is not merely additive. It's a multiplicative force, accelerating discovery and innovation at an unprecedented pace. This powerful combo of microbiology and data science cannot be overemphasized. The microbiology brings forth intricate biological questions. You are asking a question. Who are there in metagenomics? What are they doing? Who are those people that are there? We see that microbes are ubiquitous. They are present everywhere, in food systems, in soil samples, they are in water samples, in environmental samples, polluted areas, in agriculture, in plants. We are talking about host microbiome, host microbe interactions. So microbiology gives us the framework. It brings forth the biological questions with diverse samples and a deep understanding of the microbial systems, laying the groundwork for scientific inquiry. Microbiology is not obsolete. The data science role in it offers sophisticated computational analysis, powerful predictive models, ability to extract meaningful insights from complex microbial data set. You can imagine me working with about 9 million base pair metagenomic sample that contains different microorganisms. In fact, recently, I sequenced a particular isolate. I sent it out of Nigeria to a particular sequencing facility named with TED for this presentation. And it was meant to be a whole genome sequence. And when the result came out, it came out excessively large. The data, the file was extremely larger than what it ought to be for a pure sample. And I began to look at that data. I was expecting the whole genome sequence data around 2.2 to 2.3 million base pair. And what was turned in was about 9 million base pair. I look at that data, we began analysis on it, and that data turned out to be a metagenomic sample. There was contamination from the end. But you know the funny thing, there's no mistake in science. I began to delve into it, and I saw a particular isolate. You know, from that metagenomic sample, we have a lot of different mixed sample. What the metagenomics does, it tells us who are there, what are they doing, the evilness, the richness. We'll be talking about the Shannon and the Simpson index, richness, evilness. Who are there? In what proportion? Then we look at functional metagenomics. What are they doing? So data science enables us to have the ability to extract meaningful insights from complex microbial data set that you would not have been able to see in your molecular or microbiology laboratory. Accelerated innovation. Together, this microbiology and data science offer powerful combination that transcends traditional research paradigms, leading to accelerated discovery, novel solutions, and groundbreaking advancement across various fields. You know, the funny thing about this data science of a thing is that it's not limited to biology. We can analyze data for other fields, environmental scientists, chemical engineers, you know, we can analyze data for diverse walks of life, different fields of life. The same skill, can be deployed to other areas. So this interdisciplinary approach unlocks new avenues for understanding microbial communities, developing innovative biotechnologies, and tackling global challenges from health to environmental sustainability. However, some skills are required for the future microbiologists, not only the culturing, the inoculating loops, your microscope, your autoclave. Beyond that, some skills are required. I know that some things are crossing your mind that, okay, now you're telling me about data science, I'm a microbiologist. Where do I start? The good news for you is that you have to start where you are. And that's the essence of having a community like this that we have, classroom to career. The evolving landscape of microbiology demands a new blend of expertise. Future microbiologists will thrive at the intersection of traditional lab work and cutting-edge data analysis, experimental design and execution. You already have that skill. Your strong foundation in microbial culturing, molecular techniques, and microscopy remain critical for generating high-quality data. Should I amaze you? My work started from this wet laboratory, experimental design and execution. I had the idea, I wanted antimicrobial peptide producing bacteria. I want to isolate them. I want to screen them. I want to produce the antimicrobial peptides in vitro. I want to test them on pathogenic strains of microorganisms, food spoilage organism, medical related pathogenic microorganism, Pseudomonas aeruginosa, Staphylococcus aureus, Escherichia coli, Bacillus cereus, Listeria monocytogenes, and also Streptococcus pyogenes. So we have done that in vitro. You have that solid skills of experimental design and execution as a microbiologist for you to be able to generate a high quality data. The next thing you need is your statistical and computational thinking, because when you finish in our microbiological laboratory, you migrate to the molecular laboratory. Okay, now you have a pure culture, pure strains, arsenic culture. I hope you remember that in microbiology, sterility cannot be used in a relative sense. An object, an item is either sterile or non-sterile. There is nothing like semi-sterile or almost sterile. So that your skill, your arsenic skill that you already have is a foundation for you to be able to generate a high quality data. Because if you have contaminated samples, molecular biology is garbage in, garbage out. Sequencing is garbage in, garbage out. Whatsoever you input is what you get. So the quality of your microbiological data must not be compromised. So microbiology is not becoming obsolete. In fact, you need to have the sound knowledge of how your organism is. Who are they? Like me, I work with facultative anaerobes or microaerophilic organisms. So when I had a contamination, when they had contamination in the laboratory that I sent it out, I was able to query them that what I worked with was a facultative anaerobe and microaerophilic organism that can never cohabit with a straight aerobe. In fact, that straight aerobe can never contaminate my sample, not to talk of dominating the sample. But remember, imagine if I did not have a microbiology background, I would have been lost. I would have taken whatsoever they gave to me. But I was able to, make them realize scientifically that it's not plausible for an anaerobe to contaminate a sample where we had only facultative anaerobe and microaerophilic sample, not to talk of actually dominating at the level of the metagenomic sequencing. So your data is valid. Your skill as a microbiologist is valid. You know, your course as a microbiology is not obsolete. It's even more needed at this time, more than ever. We need to be positioned and strategically positioned. Even though I've been working at the intersection of genomics, biotechnology, and data science, I did not drop my microbiology skill. I'm a microbiologist. In fact, the first thing I write, when you look at everywhere, maybe on my ResearchGate, LinkedIn, Google Scholar, or see this, I'm an industrial microbiologist. That's the first. That's my major. Every other skills are minor. So be proud that you're a microbiologist. Yes. So you now need to know how to migrate between your microbiology to molecular biology, another wet laboratory, where you now begin to talk about the fingerprint, the DNA fingerprint of that microorganism or the sample that you are working with. From molecular biology, now the raw data is there, the raw sequence is there, gigantic data, 9 million base pair. You are going to be having different kind of file format, FASTA format, FASTQ format. You are now looking at how do I open this? Your knowledge of statistical and computational biology and thinking now comes up. You need to develop your proficiency in statistical analysis, programming languages like Python, like R, SPSS, the genomics and bioinformatics tools for interpreting complex data sets like the BLAST, like the NCBI, the Galaxy, data visualization like ggplot, Tableau, Power BI, that's Power Business, and the machine learning basics. So how do I harness all of this? You are wondering, they are complex. Yes, they are, but don't worry. You take it step by step because Rome wasn't built in a day. All of this comes together to be able to give insights into your raw data. Otherwise, you'll be sharing those data in the CSV file or Excel format and be looking at how do I make sense of this? But a whole lot of fingerprint, insight, functional insight is embedded in those dirty, gigantic data sets. Don't bother. It's an interdisciplinary collaboration. You cannot do it alone. Your ability to communicate effectively with data scientists, computer engineers, clinicians, bioinformaticians, to be able to translate biological insights into actionable solutions is very, very important. Wherever you are now, you need collaboration. You cannot exist alone. A lot of, different professionals come together to be able to work on bioinformatics, on visual analytics, on data science. You need to activate your problem solving and critical analysis skill. You have to apply your analytical rigor to identify novel microbial patterns, predict outcomes, and innovate solutions to global health challenges. You can imagine that I was able to actually isolate, screen, produce antimicrobial peptide in vitro. I've already tested them against different pathogenic and spoilage microorganisms. I've already attested that they were broad spectrum in activity. I've already tested them for the safety. I've already assessed their safety in vitro, that I did antibiotic susceptibility testing for them. I did immunogenicity testing for them. By the time genomic data was out, that was able to analyze their genomics, I was able to see the genes coding for those ability to be able to resist pH, the ability to be able to survive at different temperatures, the ability to be able to survive the gut microbiome, the ability to be able to actually resist some antibiotics. The gene was there in their genome. How would I have done that without the knowledge of genomics and bioinformatics and data visualization? So the need for you to enhance these skills will empower you as a microbiologist to unlock deeper insight into microbial systems and contribute to transformative discovery. Because I normally say, if your research will end on the shelf, please think and think again. Does it worth it? Does it? So now you're asking that, okay, as microbiologists, what opportunities do we have? A lot of opportunities, my dear colleagues. A lot of opportunity for microbiologists in data science. Because the fusion of microbiology and data science opens up a vast landscape of career opportunities. As microbiologists, our deep understanding of biological systems, combined with data-driven approaches, allow us to tackle complex challenges and drive innovation across various sectors. You can imagine, as a microbiologist and industrial microbiologist, I'm offering solutions to food systems, I'm offering solutions to agriculture, I'm offering to medicine, I'm offering to pharma, I'm offering to public health. You can imagine. And I'm just an industrial microbiologist. I found something, a product that can serve food industries, agricultural industries, pharmaceutical industries, drug development industries, medical industries, and also even public health. I have found a solution to antimicrobial resistance that's ravaging the globe. So you have an opportunity to be a bioinformatician. You have an opportunity to go into bioinformatics and genomics research. You can leverage computational tools to analyze vast genomic and proteomic data. Should I shock you? You don't even need to go into wet laboratory. There are a lot of data set available online and secondary data that you can download and start working on immediately. For your genomics research, they are there. As long as you acknowledge those people, the authors of those sequences, you are good to go. So do we need to generate more data without analyzing them? My own case is different because I was able to take my own research from microbiology, in vitro, to molecular biology, to genomics and bioinformatics. And now here's the product. But not everybody has that prowess to be able to come from microbiology, molecular biology, to genomics, to bioinformatics. But wherever you are, my dear colleagues, you can start from where you are. You do not, if you don't have a laboratory, wet laboratory to work with, you can work at the intercept of dry laboratory, in silico. Are you aware that research can be done in vitro, in silico, in vivo, and in situ? In vitro is just what you are doing in the laboratory. In vivo is on animal and human studies. In silico, your computer, bioinformatics, visual analytics, data analytics. In situ, the one done in the environment. So you can start your research. If you don't have a laboratory to start with, you can start with an in silico research. You need partnership, you need collaboration. You can uncover insights into microbial evolution, function, and interactions. You can download some genomic data, proteomic data, and begin to analyze them and make inferences from them. This is crucial for understanding disease mechanisms and discovering new therapeutic targets. Are you aware that I told you I had a contamination from the laboratory that sequenced my data, that sequenced one of my isolates? But those data are not lost. We've started delving into it, and a lot of novel EMPs are coming out of them because that sample became a metagenomic sample, an environmental sample, that even though they were contaminants, but they embedded the sequence, the genes of novel antimicrobial peptides. We are not discarding those data. Then you can be a public health surveillance, you can work at the interest of a public health surveillance, utilize data science to track outbreaks, predict disease spread, and identify antibiotic resistance patterns. Like in my own isolate now, I saw a VANG gene, and that VANG is responsible for actually vancomycin resistance. And that corroborates what I saw in vitro. Now that isolate was able to was resistant to vancomycin. And the genomic footprint showed it in silico. So microbiologists are essential in developing early warning systems and informing public health interventions. Pharma and drug discovery. You can apply data analytics to accelerate discovery and development of new antimicrobial agents, vaccines, and biotechnological products. This involves screening massive data sets for potential candidates and optimizing their efficacy. What I have with me now, I have different biosynthetic gene cluster, big set of genes for antimicrobial peptide synthesis that we can cut into different segments to synthesize antimicrobial peptides as alternative to antimicrobials, alternative to antibiotics to combat antimicrobial resistance in the global village. As a microbiologist also, you can also work at the intersection of agricultural microbiome engineering. You're looking at plant microbe interactions. And next power of microbial community to enhance crop yield, improve soil health, and develop sustainable agricultural practices using data to optimize interventions and monitor their impacts. Food safety and biopreservation, that's one of my key areas. We employ data science to identify foodborne pathogens, predict spoilage, and develop innovative preservation methods like I'm doing, ensuring food safety and reducing waste throughout the supply chain. Challenges to adoption, bridging the divide. You are wondering, oh, see grammar, how will I start? Despite the fact that you know that we are in a low resource settings, this woman, you have started again. Hello? The good news is that we can start where we are. Integrating microbiology into data science presents unique hurdles, no doubt, particularly in resource-constrained environment like ours, LMIC, and across specialized disciplines. You are wondering, I'm a microbiologist, I don't have data science skills, I don't have genomics skills, I don't have molecular biology skills. Relax. Addressing these challenges is crucial for unlocking the full potential of this interdisciplinary field. It's not only one person's field, it's a collaborative field. Knowledge gap in programming and coding. Remember when we went on 200 level, I think they taught us programming then in school. Many microbiologists lack formal training in programming and data analysis, creating a barrier to effectively utilizing computational tools and interpreting complex data sets because Python and R, they work on coding. They don't take instructions like the Microsoft Word or the Mac. You have to actually code to be able to, give it to them, do this. They understand coding. Dedicated educational initiatives and user-friendly software development are essential. And that's why we are together. Peradventure you want to take this forward, then we have limited infrastructure in developing regions like our region, access to high-speed internet, reliable electricity, sophisticated computing resources remains a significant challenge in many developing countries. We are aware of that, hindering the ability to engage in cutting-edge data-intensive research. High cost of sequencing and computing power. One of my isolates for the WGS cost me 500,000. After I've already gone ahead to sequence about 6 of them, about 500,000 or so, then one of them that came back as a putative novel strain was sent in for WGS, another 500,000. You can imagine if this resource is not there. So high cost of sequencing and computing power. The financial investment required for large-scale genomic sequencing, because in this part of the world now, where we are, for Nigeria now, the sequencing facility that we have, I think they are private. Even sequencing is not being done here. They send it out. The closest to us, I think, is in South Africa. So the financial investment required for large-scale genomic sequencing and the computational power needed for big data analysis can be prohibitive for many research institutions, especially smaller ones. Not to talk about actually early career researchers like you and I. The need for interdisciplinary collaboration is one of the challenges. Effective research at the intersection of microbiology and data science demands seamless collaboration between experts from diverse fields. Fostering environments that encourage cross-disciplinary communication and shared understanding is essential. And that's why we are together on this forum. Future outlook, data-driven microbiology. What does the future holds for us? I can tell you, my dear colleagues, that the integration of data science into microbiology is not just a trend. All of us have to come to terms with it. It's the future of innovation and discovery in biological sciences. You can imagine if I have stopped my research at the level of that in vitro analysis. I didn't go to molecular. I did not go into genomics and bioinformatics. How would I have been able to predict the genes coding for the antimicrobial peptide that I've seen now as alternative to antibiotics, in agricultural, food systems, medical, pharmaceutical applications? The future of personalized medicine is here, right in your hand. And the good thing about microbiology is that they can't take us away from this. We are the center, at the forefront of this. Tailoring antimicrobial treatment based on individual microbial profiles. Environmental bioremediation, because you can look at human being, you can look at the genome of human beings and look at the gene coding for this AMR resistance and check it whether it's present in the human being, in the particular human's genome, and know which of those antibiotics that will be able to work for that person and customize antibiotics for individuals. That's the future that we are looking at. Personalized medicine. We are looking at environmental bioremediation, developing microbial solutions for pollution control and ecosystem restoration. Synthetic biology, designing novel biological systems with enhanced functions for various applications. Global health security, rapid identification and response to emerging microbial threats and pandemics. When the coronavirus actually happened, they were able to respond fast. Why? Because of genomics. They were able to actually sequence the genome of COVID-19. They saw the way it's actually metamorphosed and actually mutate fast. They were able to catch up with it. And there's one I would like to add into this: food systems, biopreservative. We are able to use data-driven approaches to predict biopreservatives, alternative to synthetic chemicals and antimicrobials in food systems. Join the revolution, my dear colleagues. I charge you this evening to join this revolution, because if you don't catch up with it, we are already going ahead. We are not waiting. Collaborate and innovate. Microbiology plus data science is the future. Microbiology is not obsolete, it's evolving. We have to be positioned for it. Data science is the new language of biology. Alumni and young scientists must embrace this future, otherwise we will leave you behind. Explore the vast potential at the intersection of both fields. Collaborate to drive groundbreaking discoveries. Shape solutions that benefit humanity. I tell you again, if your research is going to end on the shelf, think, think, and rethink. And I'm asking you now, if you want to ask, if you have any questions, want to reach out to us for further discussion and collaboration, kindly connect with us on LinkedIn, connect with us on ResearchGate, connect with us on Google Scholar. Together, dear colleagues, we can redefine the boundaries of biological innovation. Thank you for being part of this journey where microbiology meets data science. Thank you so much, Dr. Monyi, for giving me this opportunity to speak to our community. Thank you, and I remain yours, Atinuke Pokuola.
Okay, thank you very much. We are grateful for your very sound presentation and for giving us so much insight on data science, and especially what you do also, because it's amazing. And I think that it's come up severally, especially in the place of skilling up, where we have to where we should think of how to add more skills to ourselves. We need to think of how we can learn these new skills. For instance, how to use Python, how to use R, even as SPSS, because I think a lot of people, we run away from statistical analysis, and I believe that. I also sometimes look at, why do I have to do statistical analysis? But it's very important. So, if we have any questions, I think we can please raise our hands or unmute ourselves and ask questions, or type in the comment box your questions for our speaker so she can answer your questions. Please, do we have anybody with questions? Anybody with questions? If you want to learn how to do something, how to use a particular software, data analysis software. I know a lot of us find genomics very complex sometimes. Yes, I myself, sometimes I'm like, oh, why do we really need to do this? Or why do I have to do this? It's genomics. I don't feel like doing it. It's very complex, and so if there's anyone that wants to talk to us about it should please go ahead, ask your questions, and she can answer it before we round up.
Hello, can you hear me?
Yes, we can. Yes, we can.
So my question is, what's the best way to teach yourself like R and Python?
Over to
Did you
Yes,
you.
hear me?
Yes.
yes, I can hear you. Thank you so much, Dennis. Yeah, you asked a very beautiful question. That was the best way to teach yourself. Okay, if I may tell you, I think the first time I was exposed to visual analytics was in 2012. I was the first project manager of H3 about net, NABDA node, that's Human Heredity and Net in Africa, for the first 5 years, 2012 to 2017. So at that time, I was fascinated with visual analytics, data analytics, bioinformatics, and all of that. But, until you have your data to work on, the zeal might not come, because it's not arbitrary. It's not just like vague. There must be something you are working with. There must be something that is driving you, your passion, your interest. Because if as a biological scientist, we did not go in depth into coding when we were in school, but I knew we did a course that we actually we were coding. If I remember that course now, not MAT 211. I think it was MAT 212 or so, second semester 200 level. Now we went to computer science to I think Fortran, formula translation. Yes, I can actually remember that course. That course is like uh is like the course is a basic, you know. So when you have a data at hand to analyze, begin to wonder, which interface do you want? Do you want R? Do you want Python? For statistics, R is, for if you have a data that you want to analyze, you have we have descriptive, and we also have qualitative data, quantitative and qualitative. So for data analysis, like statistics, like SPSS now, they do data analysis for quantitative. But if you want visual analytics, R has all, both descriptive, both qualitative and quantitative data. So the best way to teach yourself, no matter you have been exposed to like this kind of webinar that we have now, do you have a data, if I may ask you to analyze? What kind of data do you have? What are your interests? You need to have a data. Then you learn to read. You can take a free pace, self-paced course on Coursera, like all those introduction to R and all of that. So they introduce you to different data structures. Because even the way you package your data, you need to actually learn different kind of data structure that you have. Even the way we name in data cleaning. For instance, now you have an Excel sheet, very wide Excel sheet. You want to say name of students, name of pupils, and you are writing name, space, of, space, pupils. R will not recognize it. It will not recognize it. There's no space. You either use name underscore of underscore pupil. You are trying to connect those data, even though you are segmenting them. Their name, of, and pupil are not the same, but you are using name underscore of underscore pupil. That's data cleaning. Like today, before I joined this webinar, I had a data somebody sent to me to analyze. I was working on that data. I was already writing code to put on R. Then the output was not coming out. About 2 columns were left out. I was now wondering that why was these columns left out? I had to go and look at that data, look at the data that was sent to me, the raw data. How were they processed? There were gaps in the columns. I had to do data cleaning first before putting it on the R. So the best method to learn R by yourself or Python is, number one, do you have a data to work with? It must be a data that you want to work with to start with, no matter how small that data is. Number two, you need to go for self-paced courses, or you paid for courses. Some people normally pay to teach people. Maybe 1 week, 2 weeks, 1 month, 2 months, 3 months. You need to learn. And before you know what's happening, initially you might not actually be proficient. It doesn't matter. Don't worry. You are getting there. Even me myself, even though I've been exposed to data analysis for the past so many years now, when I was doing my own PhD, I was thinking I was working too much in the laboratory. I was also writing too much. I engaged somebody. Let me burst your bubbles. The person had OND statistics, HND statistics, BSc statistics, MSc statistics, and PhD statistics. An HOD in a particular uni, sorry, a HOD of statistics department in a particular uni in the Southwest. I will not mention the school. Name with it. I paid him since December last year. June this year, did not turn in my results. I gave him 21 data sets. I was thinking that because I was having too much on my desk, let me contrast this out. I paid. Coding for 21 data sets. I was thinking that to take my time, and I had a lot of things to do. It didn't turn in my data, and I've paid. So I was very angry in July. I think July 10th, I called him. I said, please stop. Give me back my money. I'm not doing it again. I want to give another person. My brethren, I have to go back to my R. But to the glory of God, I analyzed all my PhD work by myself. When I made my final presentation, I think some of my colleagues, and I don't know, maybe they are connected here, they can attest to the fact of what happened. They were asking me in school, please, who did your statistical analysis for you? Because my chart was something else. I went back, even though I didn't sleep 24 hours straight in many days. I would just sit down like this from beginning, from morning till evening. Another day will come. I will open my eyes. I'm on the computer, analyzing data, coding, troubleshooting, you know. So the best way to teach yourself is to join forum like this. You have already been exposed to it. The next thing, make another step by probably going to learn self-paced, or you want to pay to learn. Because if you want to pay, my brother, they will not, it's not free, because they did not also learn it free. That's the truth. You can also go for Coursera, go and look for R package in Coursera. You can go for that. Then first, generate data to use. I have large data sets. You can imagine I said the first data I gave that man was 21 data sets. Did you hear that? And I had more. You can imagine the one that could not analyze 21 data set. What did the one that, and I delivered 4 manuscripts within 18 days. My lecturers were actually opening their mouths. How did you do it? And not quick journal, Q1, Q2. The first one dropped day 1, day 5, day 10, and day 18. Days, 24 hours, I was not sleeping. I won't see sleep, coding and troubleshooting. So what am I saying? At the point, my lecturer asked me, my first supervisor, that, sorry, from this manuscript, your data, did you add other data to it from your thesis? I smiled. I said, no. If my thesis appear like tables, when I'm sending it from manuscript, I won't send a table. I will send it its map, or I send another plots, line graph, or bar plots. She was amazed. Because even when you're doing plagiarism check, like somebody now in my team has an issue. She just copied all her thesis, dump it on one journal. Unfortunately for her, the journal came out before she submitted her thesis. Her thesis now is already, it's red. But it picked self-plagiarism. Everything on that manuscript was actually flagged in her thesis. These are skills that we learn. So it's not free. So if you want to actually learn, the best method for you to learn, what are you working on currently? What kind of data do you have? Let's start from there. You know, then do you want to actually learn from someone, or you want to do a self-paced learning, which is not so bad. You can actually have your data, go online, go and take some courses, and you begin from there. And before you know what's happening, you are mastering your skills. And I can tell you that it can only get better. Thank you so much, Mr. Aden. Do I answer your question?
Yes, yes, yes. Well answered.
All right,
So much.
thank you. Does anybody have any questions? So, Dennis she says you have to master your skills. I think it's very important that mastering skills and learning is very, very important, especially with data science. Without your data, you really cannot do. If you go for a course, and that's the truth, if you go for a course today, or you go to somebody to teach you, if you don't have data to work with, you will not learn it. So I believe with data science, you have to keep doing and doing and doing over and over again so that you learn and it to get better. So that's nice. So I hope that we have learned something today. And if there are any questions, we're going to send, you can write the questions, send it as email along the line. You also, we would also make the recordings available. We'll make the recordings available. From our last webinar, we have a YouTube page where we uploaded the recordings. So you can go also and watch and learn from it. Also, we'll make this recording available, and you can also get her contact details also. She's on LinkedIn, she's on ResearchGate, she's on Google Scholar, and we would make the presentation also available on YouTube so you can join it and watch. Oh, we have a question, Sakwe Michael. Please, can you go ahead and ask your question?
Okay, madam. Thank you very much, Doc, for your presentation. I'm grateful. I've been following all through along LinkedIn, and I'm impressed with your work. And I'm saying that you have really inspired me a lot with respect to what you are doing. So, my question is with respect to the fact that students who have taken microbiology in undergraduate and they want to like venture in other fields. Let me say, for example, biological engineering, or with keen interest in development of medical devices. How can they easily integrate such field? Should they begin in taking courses that are respect to the field that they want to integrate, or they can as well enroll to those programs? If, for example, the program are not offered in their country but another country, should they enroll immediately, or they should first take formal courses like self-paced before enrolling? So that's my question. And also, are there available resources that you can as well, let me say, for example, like a textbook or an article that you can refer us to, that we can easily go and learn some of these data science skills? Thank you, madam.
Yes. Thank you so much, Mr. Sakwe Michael. I'm so happy to have you here because I know that name rings a bell. Any small thing, he has liked the linking post, he has tagged me. I should look at this. So that name is so familiar. Mr. Sakwe, thank you so much for joining. Yeah, if I may understand your question very well, there's some undergraduate students studying microbiology, but they have interest in developing some devices. In fact, that rings a bell. I remember a project recently that I think I wanted to collaborate with one HOD in a particular university in the southwestern Nigeria, involves development of a biosensor. And I knew that we've written some books, I think, is it a Severe now or Springer, on biosensors, you know. So the good thing, like I said, microbiology, biological science, we are not mishaps. In fact, we are blessings because those biosensors, we are the ones that will detect them. Those equipment or devices, they are just like carriers. If I remember, I think it's a tuberculosis sensor that we wanted to do from urine rather than sputum. The current one available now is, I think it's from sputum. We're trying to prefer the one that is not invasive, that somebody can actually can detect some of those antigens in the urine. So we needed to bring on board a biomedical engineer. No, the science has gone wide to the point that only one person doesn't rack brain any longer. We do most of our research work collaboratively, interdisciplinary. As a microbiology, learn your microbiology. I tell you something, I am standing as a microbiology. Anywhere I am, give it to us. Our course is not obsolete. It's a foundation. And that's why wherever I am, I did not drop microbiology. So I'm a biotechnologist, or I'm a molecular biologist, or I'm a bioinformatician, or I'm a data scientist. I am an industrial microbiologist. And I'm glad and I'm proud to be a microbiologist. So to answer your question, if you are trying to develop probably a biosensor or a device, they don't need to stop microbiology. Let them continue, because the microbiology will give them the basis to be able to understand antigen antibody complex. We did one course, I think immunology, I think MTB 301. I have Olubumi first one on this platform. I don't know, maybe it's the husband that's using the phone or is the wife. We were together in microbiology department, University of Ado Ekiti. We did a course in biochemistry. Is it biochemistry or microbiology? We did I think immunology, I mean, and immunochemistry. Was it MCB 302 or 304? 4 unit course.
Immunology is always a 300 level. Yes.
Yes, I think MCB 302, MCB 304. I think I'm seeing Luabumi first one here. So we did that course on antigen antibody complex. That will enable you to be able to know even what antigen to look out for, and what antibody are you bringing in. Antibody is from the inside. Antigen is that thing that will simulate it from outside. That will not we have inflammation or all of those things. The way they normally talk about it when we're there. So let those people continue with microbiology to give them a background, of what microbiology is all about. For development of a biosensor or a device, what you just need to bring a biomedical engineer into that team. You need collaboration. You cannot do it alone. They can take a course to understand, but they cannot totally, because biomedical engineering is a 5 years course. I think they have it in University of Saskatchewan. Maybe in Nigeria, some schools are also coming up with it. In Nigeria too. I was looking for a biomedical engineer on LinkedIn when we were developing that project. I think I got one. Even they were telling me that they didn't really have hands-on experience as such. And I brought somebody again from the US too, whose hands are full. So I appeal to you, Mr. Sakwe Michael, let those students continue microbiology. We only need a forum like this to open people's eyes. The microbiology is not becoming obsolete. We only need to lace it. We only need to actually package it. Like we set an awareness now to let the schools know that there's a need for them to integrate a little bit of biotechnology into microbiology. But for them to actually quit microbiology and go and study biomedical engineering, no. Because I can tell you that those biomedical engineers, they are still lacking some skills that we have in microbiology. They will not understand the nitty-gritty of those antigen-antibody complex. They only want us to, like I told the guy in our team, that biomedical engineer, I explained to him that what we need to do, we have the antibody, we have the antigen. Can you help us place this as biosensor, in a casing? So what they need to do, electronics engineer, biomedical engineers, is we are the one that we did. Look at insulin, for instance. Look at the sugar test, that test strip. Those tests were developed by biological scientists. But the casing, maybe biomedical engineers, we come up with that equipment, that small device for testing blood sugar. So my brother, Mr. Sakwe Michael, those children, they're on the right path. Let them continue. In fact, if you don't mind in your team, get a biomedical engineer as an expert. When you get to the level, you are going to be the ones to develop what you want. They will just help you to put those biosensors in a casing, and that's all. So most of the time, we work at the level of integration, at the level of collaboration and partnerships. Then for hands-on textbooks and on R and all of that, a lot of them are available. No, now we have democratization of information online. We have a lot of courses on Coursera. There's out there, I know, learning of R. And there's some schools, if on this LinkedIn, I think University of Stellenbosch, I'm the alumnus too. I actually took a course, a 1-week course there too. There's one man, I can't pronounce his name now, but I think after this class, I can actually give you a link. Every year, that guy normally teaches Python. I was there for 1 week. Even though I've been exposed to R, I've been exposed to mathematics since 2012. But anytime I had the opportunity to refresh, to rejig, I wouldn't mind. If I have the time, I'll put in for it. So for 1 week, I locked down. I was with that guy. The guy is good. He has YouTube channel, a lot of resources on his channel on YouTube. University of Stellenbosch. After this class, I think I will share the details with you. Go and follow that guy up. So and for instance, for data, for instance, like we're asking Mr. Dennis, do you have your data? You can start by downloading data from online. NCBI has a lot of data. Uniprot, Assemble, they have a lot of data that you can download and start practicing with. Do you understand? Even ordinary data that you have in a laboratory in a Z sheet, we can start from as small as it is. We can start from it. And if you want us to actually have a small class for R, we can do that also and begin to form
And
a,
so she
you
has
know,
time now. So she has defended PhD so she can teach
who
us.
has time? But you know, anything for the community. So
I
we can
am.
actually, okay,
You want
somebody,
the class,
It's
right?
a coincidence that I am from Stellenbosch.
Aha.
can you see? Are you serious? You're from Stanley, but there's a guy. I can't Rasta something. I will bring up his name. I don't want to pronounce. I don't want to misspell his name. I've been following that man for I think in 2020 or 2021. I took a class. Was it in 2020 or 2021 in Stellenbosch? So when I'm saying thank you to University of Stellenbosch, Hunter College, University of Mississippi, and all of that, those are my people that I've actually eaten and have taken from over the years. It's not a one-day thing. But we can start from somewhere. And I can tell you, when you have somebody that can hold your hand like we are doing, to open your eyes, to tell you exactly the way to go, you won't have problems. And the job becomes easier for you. Like for instance, now when I think it was that last Thursday that I did my finals for my PhD, somebody told me that okay, you sent somebody to me that they needed help and all of that. And when the person came, I was thinking that they wanted me to help them to analyze their data, but they were saying that I should come and teach her how to code. I said, sorry, your coding is not a one-day thing. You can go and take a self-paced course because as I have now, a lot of things on my desk. I was thinking that you wanted to send the data to me. Let me quickly help you run it and you have your result and go on. If you should learn coding, for instance, I want to ask you that, okay, let's go for when we're learning R, we were taught with Linux. We partitioned our laptop into two. One was Microsoft, the other one was Linux. So set working directory, you start from there. Get working directory, you start from there. So let's now start teaching you how to start putting set working directory, get working directory on Linux. That for now, that time is not really it's not available for me, but you can go at a self pace and go and do. But however, going forward, we can actually start from somewhere. What kind of data do you have currently? Let's start from where you are. Everybody, go and check your laptop. What are the kinds of data that you have? No matter how, as I am now, I make sense of every data. I don't discard data. Every data is very, very important to me. I told you I have, from my sequence, whole genome sequence, they brought me metagenomics data. I was expecting 2.2 million base pair. They sent me 9 point something million base pair. Even though I challenged the sequencing authority, but those data, I'm not going to discard them. I'm making sense of them. So what data do you have currently that we can start with? Let's leverage on that data. See Mr. Deji, my colleague. Doctor, God bless you, sir. They follow me here. Doctor, I'm happy to see you. Thank you so much for joining. So let's start from where we are.
I'm in the vehicle. Thank you very much, ma.
Oh, thank you, Doctor. God bless you for joining. Thank you, sir. Yeah. So what kind of data do you have? Let's start from there and leverage and form a formidable bond. You cannot do it alone. If you are thinking that you want to be the only biomedical engineer, you want to be the mathematician, you want to be the microbiologist, you'll be way down. You need collaboration, you need integration. Like for instance, when I was analyzing my data, I had to employ a company, a genomics company. We were looking at those data together. I paid in dollars, you know. So I had to pay because it's a service. We were looking at it together and we were able to bring insight. That sequencing company had to call me and say, please, what kind of database was that? Because I was sharing the report with them. What kind of database? I now say, well, it's not a database, it's a visualization tool. They've not seen it before. So nobody knows it all. We need collaboration, we need integration, we need to come together and form a formidable bond amidst low resource that we have in the LM high C region. It should not deter us. We got to move. So I don't know if I'll be able to answer the question of Mr. Sakwe.
Yes,
Mr.
yes,
Sakwe,
madam.
I will send you a link to that man, Minister of Stellenbosch. The guy is an authority in Python.
Okay.
Yes, he has a lot of YouTube videos that they can watch. But even aside from the watching of the videos, we have to practicalize and use some data to be able to learn one or two things. The more we
Okay, madam.
practice, the more you are proficient in it.
Okay, Doc. Thank you very much.
You're welcome.
Because with respect to the fact for the biological engineering, you made mention of the biosensor aspect of it. and that is really part of one of my research interests. Uh, since I am in my final year postgraduate studies, studying microbiology, so I'm particularly interested in working with to develop an immunosensor to detect bacterial contamination of water samples or water around different vicinities in my locality. So I don't know. I contacted one of my seniors ahead of me to help me out with this project. So I don't know his function is at the Department of Electrical and Electronic Engineering. I don't know if there is possibility that both of us can collaborate with this work on this work to see that it comes to reality.
I think
If I
it's
can work
okay.
with him.
You
Okay,
should
that
be
who can
able to
collaborate?
collaborate. You want
The
to collaborate
who?
with you and that
Okay.
colleague, yes.
Okay. So is this your master's
Masters.
project or your side project?
It is my project that I want to work with this final year.
Your
So I don't know.
master's project.
Is it masters or PhD?
Yes, a master's project. So far, so good. I've not. I suggest it to my supervisor or my supposed to be supervisor.
Okay.
He said I should go and look more insight on the topic, like to read
How
some
many months
articles.
is the project? How many months is your project semester?
It is respect to the student or who is supposed to carry
Okay,
out the
at
project.
your pace.
Yes,
Okay,
yes.
where are you? Are you in
So, but,
Ghana? Where are
but
you currently?
I'm based in Cameroon, Boya
Cameroon. Okay.
University of.
Yeah, what you just said now is doable. You know, that's one thing about experience. For instance, I'm an industrial microbiologist, but to the glory of God, I can tell you that I am vast. Like this biosensor, I know we've written a book project on it. I knew that I wrote. I think my own chapter was detection of E. coli. I've written about biosensor. I know I can pull up those book chapters and see. I think is it on a Severe or Springer? I can't remember
Exactly.
now.
I'm also keen interest with the E. coli aspect. I read
Like
an article on development of biocell of a plastic immunosensor, optic fiber plastic
Optic
immunosensor.
fiber. So you have to think of the materials you have to combine together to make the base for your matrix for your biosensor and how to so you look at it from different angles, whether you are looking at the what you are the biological agent, the transducers and whatever you're using to make the sensor. So it's a very broad work actually. And I think if you can do it as a self-space, you work with her, with our latest Dr. Pokwala, you work with her, you work with a lot of people, do you understand? I think it's also good. For instance, I also I'm looking at actually doing something on that from the angle of material science also. I work in the intersection of microbiology and material science. So it's nice actually. So there are a lot of factors to consider. So I think you can reach out to Dr. Kupola. She would be willing to help you, or she'll be glad to help you. And she should be able to maybe connect a lot of people together to make this work for you. I don't know if you are really into it, then I think you can get it to work. If you are really into it and you really want to do it, I think you can. Don't be discouraged. I think
Yes,
and I also don't think
I'm
you
really
don't need
interested
to.
in the
I
topic.
don't think you
Just
need
that
to take any course.
the duration also, because actually we are to defend in the month of July. So we
Next
have
year.
from now, yeah, next year, we have from now to prepare, let me say, the proposal and start the wet lab work till July, let me say, because we are going to submit the dissertation work in the month of June, entering June. So if the plan of action shows that we are to begin in November, then by April we should be done with everything. Let me say both the lab work and they begin the writing in April, and by May ending June we are
It's
done with the
very.
writing.
So, but
Yes.
you have
So
you done your literature review on that? So that's very important. You have to know, you want to work with E. coli, environmental water detection and all that. You have to do like your deep literature review so that with a deep literature review, it makes it easy for you to move fast. If you don't have a deep literature review, then also is your super, because it's the one thing to really want to connect with people, is another thing for your potential supervisor to want that connection. Do you understand? So
Yes, madam.
it's difficult. So you also have to have a supervisor that is willing to want Dr. Popola to sort of co-supervise you also, that kind of thing, because of our expertise and all that. So those are the factors you really have to sit to think and look at critically. And if your supervisor is somebody that is open-minded and willing, I think that the collaboration is going to be very perfect for you.
Okay, madam. Thank you very much.
Yes, I also think that at master's level, well, most of the time they are time bound because in Africa, so that you don't elongate your time, especially if we are looking at it as an exploratory research, we are just trying to, like if the one I told you about the tuberculosis biosensor, we are still on it too. So that it doesn't take your time. I think for masters, it's good for you to work within the limit of what is achievable, because for every research, the goals must be smart, specific, measurable, achievable, realistic, and time bound. And because master's project is time bound, so and now we are going into an explorative, even though you have interest, you have passion for it. But at the same time, passion alone cannot actually sustain your masters. You must finish when your mates are finished, except if you don't mind that you really want to put your head and get it done. Otherwise, this research can be done even after your masters. So can you work within the limit of what is achievable at that level? Because some professors not like mine will not allow you to bring in external perspective. You already have a way of doing it, maybe probably two from within, one from outside. So once that 3 slots is actually logged, he doesn't want to hear about any other person that is coming into that board. So because now we need to bring on board, like you said, that electrical and electronics person, they can do it. Once from your own side, you're able to get your antigen and your antibody. We look at that autoimmune disorder that me is talking about. After this class, we can talk about it and we look at what it entails. Can we get them? If we can get them, it's achievable. And then the electrical and electronics person can be able to help us to couple it together. That's what they are doing. So that's why I said, as a microbiologist, as a biological scientist, you don't need to rack your brain about biomedical engineering. It's not your work. This guy spent 5 years in school to learn coupling, to learn how to assemble, how to disassemble, and all of that. Our own is to work with biological molecules, with microorganisms, to look at those antigens, the antibodies. Where are you picking it from? From the water samples and all of those. So we have to look at it holistically and check it out that is it achievable within this, between now and that June next year that you are submitting your papers. If everything that we need are on board and that guy is ready to hit the ground running, after we have tested that biosensor, they're able to help us to couple it into what the carrier, the package, then you are good to go. So between, I think you have about maybe 7 or 8 months between now, or even 9 months thereabouts. So let's see what is workable. But if it's not achievable at the level of masters, we are not going to discard it. It's an idea that must be born. So far you have conceptualized that idea, we must bring it to fusion. So I'm not saying that it's not achievable, but we have to go and look at it. How realistic is it? And we hit the ground running with it. So thank you so much, Mr. Sakwel Michael.
Thank you very much, madam. What you have just said has really cool down my aspiration with respect to that aspect, and also giving me a better path on what I can venture into with respect to my postgraduate study now. As you said, it's something that even we cannot achieve it now, we can achieve it in the future run. Yeah,
Yes,
thank you very much, madam.
you're
I've
welcome.
taken keen note of that. I'm
Yes.
grateful.
So we are
All
not
right.
discarding it. We just have to go and look at it and look at literature, look at what we need. Can we get them? That other electrical engineer, is it ready for us? So that once we are able to do the biological aspect of it, can they help us put it together? What they are just doing for us is packaging. Do you understand? So we are the
Yes,
one
madam.
behind the science. That's why I don't hire my biomedical engineering. That what they just did for us is to package. We are the owner, we are actually the custodian of those biological molecules. We are the owners of those antigen antibody complex. We are the owners of all of those things that we are bringing out. Do you understand? Their own is just,
Yes,
we
sir.
have found this. Can you help us put it in a portable device? That's what they are doing. And the electrical person will know that, okay, there's battery in of this. Is this solar battery that is power is chargeable, like all those sugar test kits, and the device, just portable device. They are helping us to now do a biosensor to be able to detect E. coli in water samples. That's what you want to do. So let's look at it and see whether it's achievable. If it's okay, and if it's achievable, then we strike it and we go ahead. But if it's not achievable within this confine of your time, then let's put it in the loop. While you quickly run with your master's project that you can deliver and submit, then we run with this one that you're talking about. Thank you so much.
Yeah, so thank you very much, Ma. We are grateful. I will wrap up with this thing, and I it's just a short story of something that happened to me when Doctor was talking about microbiology is the backbone for a lot of things. I remember when I did my PhD defense, my external examiner asked me, because I was working on production of a biopolymer is a bacterial polymer. And he asked me a question. He said, you have done a lot of things with polymers, polymer analysis, polymer science, and all that. He says, how many percentage of your work is microbiology? You know, you are doing a PhD defense, you are already maybe a bit agitated and all that. And I was like, 60%, 50%. I just said it like casually, do you understand? And he told me something. He said, never, never say that. If you take out bacteria from your work, if you remove the bacteria producing that compound or that polymer from your work, then you don't have a PhD thesis. At that point, it dawned on me that really, even if you are doing anything as a biological scientist or microbiologist, if you remove that bacteria from that work, there's no bio in it anymore. So we really have the foundation and the backbone for a lot of things. So it's just a way to close and say that let's be proud of what we are doing, and we function in almost every field and every possible field of study, and we are very good to collaborate with. So I remember from our last session we had a list of resources of how you can skill up and get more skills. Our speaker also mentioned on Coursera and all that. So we still have that document. We can also send it out to those that need it now. You can send an email to us if you need it. We can send it email to you with the list of courses, things you can learn, and how you can skill up. So, and I believe that we've had a good time today, and I'm grateful to everyone that joined. So if you have any questions, please don't hesitate to write us an email. We are going to also send a summary of what we talked about today to everybody via email so that you can go through it and read through it again. We will also send the links of the YouTube when we upload it so that you can go back and watch and watch and watch so that you have understanding. I know there's so much to take in at once, so you still have time to take in more at your own pace. So thank you so much for joining. I really appreciate it, and I hope that we've all learned something and we are all willing to go the step further and also learn more and build on our learning experiences. So thank you, everyone. We are grateful. So we have some information also in the chat. We would find it and send it to you as you need it. Our speaker's LinkedIn profile is also here. You can connect with her on LinkedIn and also get as much information. You can reach out to her. You can reach out to me also on LinkedIn anytime for questions or send emails. We would be available and will be willing to help and guide you as you go on your journey. Thank you, everyone. We are grateful. Have a good night rest. Thank you and bye.
Thank you.
Thank you.
Thank you so much for bringing me on board. Thank you, everyone that joined. It's a pleasure meeting every one of you. Yes, thank you.